On one day in 1952, Robert Briggs and Thomas King were working on a technique that would spur science fiction for years to come. They took an embryonic cell and destroyed the nucleus, then replaced it with another. This zygote would eventually grow into an exact replica of its cell parent. This sparked scientists' curiosity, and 6 years later they would be using adult cells to create exact genetic replicas of frogs. Well, we have come a long way since that time, with clones of mice, chickens, and cows all being reproduced from a test tube. Then a new technique came available. An udder cell from an adult sheep was put into the nucleus of an egg that had its own DNA wiped clean. Then it was implanted into an unrelated mother sheep. And on the 276th try, a successful birth occurred. The proud scientist's father named her Dolly, saying that as she was taken from an udder, she should be named after the great Dolly Parton. Since then, scientists have made clones of some very specific animals. In 2005, South Korea began creating an army of genetically identical clones from their best drug sniffer dog. While only a third of dogs can pass the drug sniffer dog exam, all the clones passed with flying colors. In 2011, China and Argentina unveiled newly cloned cows with human genes inserted into their DNA. This made them able to produce human milk. In 2012, the International Equestrian Federation in Switzerland announced that cloned horses would be allowed to enter the Olympics. While there are discussions about cloning prized racehorses and perfectly marbled beef cattle, perhaps this technology could be harnessed in a different way. Dinosaurs. Well, maybe not so fast. De-Extinction cloning is closer than ever to bringing back animals that went extinct recently, have a habitat still available, and will help the environment ecologically. The thylacine, or Tasmanian tiger, was last officially seen in 1936. The thylacine is a marsupial predator that looks like a cross between a wolf and a cat. They disappeared from Australia roughly 3,000 years ago, but remained on the island of Tasmania. The thylacine enjoyed the taste of emu, wallaby, and the occasional chicken or sheep. The latter brought it into conflict with the British settlers on the island. In 1888, bounties were put on the heads of thylacines, and this, combined with introduction of dogs and habitat destruction, pushed the thylacine to extinction. But its habitat is still there and currently destabilized due to losing its top predator, making it an excellent candidate for de-extinction. In 1999, the Australian Museum started the thylacine cloning project. They dusted off the preserved thylacine joeys and began extracting DNA. When they encountered a usable strand of DNA in a joey that had been preserved in 1909, they attached an illuminating molecule to it and inserted it into a mouse embryo. This made the DNA glow blue where it was changing the mouse. This was a huge boost of morale to the scientists, who had started to lose hope until this discovery in 2010. The Pyrenean ibex may be the only animal to have ever went extinct twice. But how does a species die twice? In January 2000, Celia, the last wild Pyrenean ibex, was found by rangers in the Spanish National Park under a fallen tree. The species was once so abundant that pictures of ibexes in caves crossed from Spain into Israel. The ibex had a history of 30,000 years and differentiated into the Pyrenean ibex 18,000 years ago. But what could make a species that was once so abundant disappear? Well, nobody really knows, but several theories are available. Overhunting, overgrazing domesticated animals, and introduced disease. Conservationists knew they needed to act fast. They found Celia and took tissue samples, not knowing she would be dead just a year later. And so began the revival of the Ibex. Scientists decided to see if they could bring a viable female into the world regardless. The Ibex still had a habitat, and the Pyrenees Mountains would be better for its return. They took a goat egg, erased the DNA, and inserted Celia's DNA, creating 500 embryos, 154 of which were inserted into goats to gestate. Only one clone survived. On July 30th, 2003, Celia 2.0 was born, resurrecting the species for several minutes before tragically passing away with fatal lung defects. Scientists had expected this to be easier than cloning other animals, since goats are fairly well studied. But surprisingly, cloned animals have a high rate of genetic defects, up to 4%. This opens up another question that needs to be answered before we start creating our own Jurassic Park. How do we keep the genetic material from clones from degrading? After looking at all these complications, why bring back any animal at all? It's expensive. There may not be a habitat. The answer to this question lies with the most famous de-extinction project, the woolly mammoth. Many picture the mammoth wandering a frozen landscape and being hunted by man in a desolate and unforgiving environment. But what if I told you that the harsh, barren, and blisteringly cold tundra was once a beautiful grassland ecosystem? This is what the creators of an experimental natural area called Plasticine Park believe. But how do you turn this area into a grassland? Well, that is simple. Poop. This natural fertilizer, I dare say mammoth proportions of it, will fertilize the land, promoting grassland-type growth and add nutrients into the soil. This will convert the flora, ushering in higher biodiversity and abundance. And that's not all. Heavy mammoths thumping around on top will compact the ground, enabling permafrost to freeze more efficiently. The grasses will then act as a sort of photosynthesizing insulation, preventing melting and preventing release of greenhouse gases. What is even more fascinating is that this harebrained scheme is working. Plasticine Park has fenced in 16 square kilometers and stocked it with large herbivores, and the grass is coming back. So not only does the mammoth have somewhere to return, it can help in the fight against climate change. There is, however, a criticism to de-extinction. Why would you expend resources on an animal that has already went extinct, when this research could save species on the brink, especially when this is already being done? In 2020, Kurt was born. Kurt may look like a normal foal, but he is a cloned Przewalski's horse from DNA frozen 40 years ago. The Przewalski's horse is believed to be the last truly wild horse and is critically endangered. They are functionally extinct in the wild, but survive in tight-knit breeding programs with the hope to revive them. And it is working. There are only 2,000 left in the world, bred from just 12 wild ancestors. Kurt will hopefully trot off to his new sanctuary when he's grown to help save his species, but he is still with his surrogate mother for now. There is also another animal that is making a comeback with the help of DNA frozen nearly 40 years ago. The black-footed ferret was declared extinct until a ranch dog trotted back home with one. The U.S. Fish and Wildlife Services quickly swooped in and scooped up the colony of 18 and took DNA samples. These 18 ferrets formed the basis of the breeding campaign that puts the estimated population at 500 today. Conservation cloning has had major successes with critically endangered populations, and these species definitely have a place and an ecosystem to return to, as they are already occupying a niche. But both programs have one very critical issue ahead: genetic diversity. It is certainly a feat to reconstruct extinct DNA from bits and pieces, such as the thylacine and the mammoth. It's more feasible, but still terribly difficult, to keep the genetic diversity that scientists deem necessary to support a stable population. This number is 500 breeding individuals, which is a far cry from the 1, 12, or 18. But scientists are looking to cross that hurdle when they come to it. For now, the scientists that are responsible for Elizabeth, the black-footed ferret, will be happy to clone Mittens for $35,000 or Fido for $50,000. Thank you for watching, and we'd like to see you here next week. Is there an animal that you believe should be a candidate for de-extinction? Comment down below.